Before we get into transcription, it's important to note that there are some distinctive differences in the processes between prokaryotic cells and eukaryotic cells. First, prokaryotes lack a membrane-bound nucleus, so their DNA is a loop that is closed and folded in a region called the nucleoid. In addition to the single circular chromosome, prokaryotes have independent plasmids which can be transmitted to other prokaryotes nearby. Remember that transcription is making a copy of a specific sequence of DNA which encodes for a gene. We don't need to replicate all of DNA. However, we do need to release the tension that holds the double-stranded DNA within the area to be copied. This region of unwinding is called transcription bubble. RNA polymerase is an important enzyme used to carry out transcription of mRNA. RNA polymerase divides the template strand, that is, the DNA strand RNA polymerase uses to build mRNA, and the non-template strand, which is the opposite DNA strand that mRNA is actually making a copy of. The non-template DNA strand is also called the coding strand, and with the exception of uracil, the mRNA matches the non-template strand when complete. The initiation site is the area where RNA polymerase binds to DNA. Any nucleotides found before the start of transcription are designated upstream nucleotides and are numbered with negatives. Any nucleotide at the site of transcription forward are called downstream nucleotides and are numbered with positives. Like the DNA polymerases, an RNA polymerase performs a template-directed synthesis in the 5' to 3' direction. But unlike DNA polymerases, RNA polymerases do not require a primer to begin the process of transcription. Since prokaryotes don't have a nucleus, transcription and translation can occur simultaneously. RNA polymerase is made of 5 subunits, but one I will emphasize is the sigma factor because it starts the process of transcription in prokaryotes. To begin initiation, a promoter region must be identified. This is where RNA polymerase binds to the template DNA. The most well-studied prokaryotic organism in terms of DNA transcription translation is E. coli, whose promoter region begins at the -35 to -10 nucleotide bases upstream from the start of transcription. Once a TAC region has been identified, RNA polymerase begins building mRNA by reading the DNA template in the 3' to 5' direction and building messenger RNA in the 5' to 3' direction. The last step of transcription is termination and can be controlled in one of 2 ways. First is by a row-dependent termination, by which a row protein follows behind RNA polymerase. Eventually, the RNA polymerase runs into a series of G nucleotides, slowing it down. This causes the row protein to catch up and collide into the RNA polymerase, knocking it off of the DNA. The second way is called row-independent termination. This is caused by mRNA folding upon itself using a non-coding strand, which causes the mRNA to create a hairpin loop. This signals the end of termination. The mechanics of prokaryotic and eukaryotic transcription are similar, but there are a few key differences. While prokaryotic DNA is transcribed can immediately be translated since the DNA is not surrounded by a nucleus, eukaryotic transcription and translation are separate processes housed in separate areas of the cell. Eukaryotic transcription takes place in a nucleus of the cell and requires some processing before it enters the cell cytoplasm. The cell cytoplasm has enzymes that can actually degrade mRNA on its way to meet a ribosome, which does translation. While prokaryotic RNA polymerases are made of 5 subunits, sigma factor being most important, eukaryotes have 3 RNA polymerases made of up to 10 subunits each. These subunits are called transcription factors. RNA polymerase I is found in the nucleolus and is tasked with building ribosomal RNA, which eventually is assembled into ribosomes, which will later be used in translation. RNA polymerase II is found in the nucleus and is primarily responsible for making messenger RNA during transcription. RNA polymerase III is also found in the nucleus and functions in the building important adapter molecules called transfer RNA, or tRNAs. These molecules deliver amino acids to ribosomes. Initiation in eukaryotes requires binding RNA polymerase II to a promoter region. However, this is much more complex than in prokaryotes. While prokaryotes utilize a sigma factor subunit on their RNA polymerase to bind it to the -35 to -10 upstream nucleotides, eukaryotic initiation requires a series of transcription factors to draw RNA polymerase II to the location on DNA to be transcribed. This happens between the -35 to -25 region in a series of nucleosides reading TATAAA or the TATA box. First, the main transcription factor, TFIID, which stands for Transcription Factor Polymerase II, with D signifying the specific type, alongside a TATA binding protein, all upon the other transcription factor polymerase II elements. This causes the RNA polymerase II to bind to the promoter region. Once a TAC region has been identified, RNA polymerase begins building mRNA by reading the DNA template in the 3' to 5' direction and building mRNA in the 5' to 3' direction, similar to how prokaryotes do transcription. Termination in eukaryotes cannot be shown here, but RNA polymerase II does not remove itself from DNA until 1,000 to 2,000 nucleotides downstream on DNA from the mRNA that was just transcribed. Technically, the mRNA produced is called pre-mRNA, as it must be processed before being sent to the ribosome outside of the nucleus. In our next video, we're going to see what RNA processing looks like and finally translation into amino acids.